In this video, we are going to discuss regarding the rabies. Rabies microbiology. So, in the introduction part, rabies is a rapid progressive acute infection, and it is a disease of CNS, affects CNS, and mainly in the human and animals, and it's transmitted through or transmitted from one another to one another from another rabid animals. So major public health problem is the fatal. It is the most fatal disease. So this is a problem with the rabies due to fatal. Moving on, rabies virus. Regarding rabies virus, rabies virus is a rod shaped enveloped virus, single stranded RNA, and there is it belong to the family Rhabdoviridae, and it has two genera. One is Lysavirus, another one is vesiculovirus. Vesiculovirus. Regarding Lysavirus, it includes rabies, Lagos bat virus, and rabies related viruses. When we look for the vesiculovirus, there is vascular stomatitis. Vesicular stomatitis and stomatic virus and related virus, and this related virus. Moving on, morphology of the virus. Regarding morphology, this virus is bullet shaped. And when you look size, it's 75 by 75 to 180 nanometer nm, and there is one end is round, and another one end is concave or planar. And it has lipoprotein envelope with a spike embedded, and there is also helical nucleocapsids. Helical nucleocapsid, single stranded RNA, negative sense RNA, negative sense RNA. When we draw image, it look like this, and there is glycoprotein. This is the glycoprotein envelope, and there is DNA, RNA. This is polymer RNA, and so moving on, and it is also lipid bilayer. It's also lipid bilayer, native sense RNA, and matrix protein. There is matrix protein. So moving on, it has glycoprotein. Glycoprotein has the function. Glycoprotein. It helps as attachment and fusion. There is M protein. M protein helps with the major structural protein. It is a major structural protein. Then there is glycoprotein G. Also glycoprotein G. It is major factor for glycoprotein G. It has major factor for responsible of virus, for virus invasiveness. Also there is pathogenicity, and there is a hemagglutinin activity. And it's a genome. Regarding genome is single stranded, non-segmented, negative sense RNA, and RNA polymerase present with, and also RNA polymerase present with the ribonucleic code, ribonucleic protein code. So moving on, animal susceptibility. Animal susceptibility. It has a wide host range. It's in fact all warm-blooded animals, including humans. Also human. Mainly for the very susceptible for the animals like fox, jackal, and wolf, and very high susceptible. It is very high. There is susceptible for rabbits, cattle, cat, hamster, raccoon, bat, and moderately susceptible with the dog, goat, sheep, horse. These are the animals which are susceptible to this virus. So there is two type of virus. So the viruses are state virus, and fixed virus. Moving on, state virus. It is freshly, which this strain is isolated in the lab and are freshly and long variable, because it has the long variable incubation period, long and variable incubation period. Also, it's produce intracytoplasmic inclusion bodies. When we move for the fixed virus, like it's when the state virus is propagated in the rabbit by the serial brain to brain passage, and it loses certain properties and loses some fixed property, some properties and become fixed virus and fixed strain, and it has no inclusion body, no multiplication in the extraneuronal tissue. It's only infect salivary gland, and multiply rapidly in incubation period is like the 4 to 6 days, and hence this strain is used for vaccination. Further, we have to again discuss regarding the differences between these two strains. So I think moving on the differences, I am drawing a table, and better to discuss in the table. Regarding this STTs is freshly already discussed, freshly isolated. Then it is isolated after the serial of intracellular passage of the rabbit or cell lines. Like isolated after the serial of intracerebral passage of rabbits and cell lines. After serial intracerebral passage. Moving on, this is produce negri body, not produce affect salivary gland, do not affect salivary gland, affect salivary gland, and I am one thing, it's also not infect. This is not infect the salivary gland. Moving on, it is pathogenic. This is not pathogenic except it is not activated properly, not pathogenic. Its incubation period 1 to 3 months. 1 to 3 months or 20 to 19 days. When you lick these 4 to 6 days only, it's produce disease. Used, it's used for the vaccine. Vaccine. Now we are moving on. Mode of transmission. So mode of transmission for the rabies virus. So mainly transmitted by the bite of rabid animals, usually dog, by the contact of the saliva of the rabid animals, like licking. If the animal is licking, like contact of saliva, contact with saliva of rabid animals, or it's also licking, or inhalation of aerosol virus by the bat cave. Inhalation of aerosols virus, or there is a transplantation of infection tissue. Example: cornea corneal transplantation. These are some method of transmission. So now moving on the pathogenesis. In the pathogenesis, first of all, if the rabid animal is infected, so its bite, or from the bite there is some receptor, like this is a muscle, and in the muscle there is a nerve fibers going in the spinal cord. This is the spinal cord, medulla pons, cerebellum, CNS, and from this when the hippocampus in the brain, and the cerebellum. Now moving on the pathogenesis. When infected animal bite to the muscle, after bite the virus is going to this receptor. There is a neuromuscular junction, NM junction, bind to this NM junction, and passes through this nerve, and passing through this nerve and go through the ascend through spinal cord, and go into the hippocampus and cerebellum, and the hippocampus that infect in this area. This virus infect. After the infection, it come in the salivary gland, and come in the salivary gland. When come in the salivary gland, and there is centrifugal aspiration. Centrifugal aspirate, like it's a spirit in the along the nerve to these organs, like salivary gland, skin, and the cornea and other organs. This is the pathogenesis of this disease. We can also discuss in the further, like there is it's multiplied locally. Viruses start replicating locally at the site of the inoculation of the muscle and tissue. Like when it's bite, it's multiply in this region. the viruses multiply mainly in this region, and after the multiplication, the viral entry in the peripheral nervous system, this peripheral nervous system, and bind with the nicotinic acetylcholine junction receptor, and this through this neurotransmitter junction, its neural speed start with the neuronal speed, and the rabies virus spread centripetally along this peripheral motor nerve by the retrograde fast axonal transport, and it is a dorsal root of ganglia of the spinal cord in this spinal cord, and after that this is ascend through the upward through the upward in the ascending tract, or the in the ascent towards the CNS. When it come to the CNS and in fact to the rapid dissemination, the virus in the most part of the CNS like hippocampus, cerebellum, and most common being site is the mental system, like it affect the mental system, hippocampus, and the cerebellum. After that, there is centrifugal speed of the disease. When this centrifugal spread the sensory and autonomic through the nerve, and it's very like salivary gland mainly, and maximum titer. And when we look for the there is like pancreas, other organs like pancreas, kidney, heart, retina, in the retina, cornea, and the skin also we can see. And after that, it's said in the saliva, and when in the saliva, rabies virus said in the saliva of the rabid animals, and which is source of the infection of the other animals in common the salivary gland, and if it is the rabid animals, so it's spread to the other animals through the saliva, and if the rabid animals bite to the other animal, and it's transmitted. Now we move on the rabies in the human. So in the next video we have discussed more regarding this. We have lots of things to discuss.